Welcome to the Everyday Journal Club. I'm Hope, a biologist with 15 years of research experience, now fortunate to be working in a Nobel Prize-winning lab. This channel has three main goals. One, to help you stay effortlessly updated on the latest scientific breakthroughs. No paywalls, no jargon overload. Two, to build a supportive community where we can exchange the most valuable experience-based research advice. Three, to share valuable strategies and concrete examples on how to achieve meaningful scientific breakthroughs. Once we reach 1,000 subscribers, I'll begin releasing content focused on the third goal. Let's go! Let's make great science together.
Welcome to the deep dive. For decades, cell biologists have been on this hunt, this relentless search.
Yeah, real cellular mystery.
Exactly. Since, 1974. How does ATP, the cell's energy money, actually get into the endoplasmic reticulum, the ER?
It's been this huge gap in our knowledge.
But get this, a massive paper in Nature, just out in 2025, seems to have cracked it. They found the transporter human SLC35B1.
And not just what it is, but how it works. And the how is just, well, completely unexpected.
Right. This stepwise translocation thing, it literally ratchets ATP inside.
It literally ratchets about 6.5 angstroms. It's wild. And it's a huge leap, really, for understanding the basics of cell biology.
So that's our mission today, isn't it? To take you through this detective story.
Exactly. We'll pull out the key findings, the surprises, and, what it all means for how cells work, maybe even for disease.
Okay, let's set the scene first. Background time. The endoplasmic reticulum, the ER, super important organelle.
Oh, absolutely central. Think protein folding, quality control, shipping things out. It's the cell's main workshop.
And all that work takes energy, right? Like a lot of energy.
Precisely. ATP. But here's the twist, the real kicker.
It can't make its own.
Nope. The ER itself can't generate ATP. It's totally dependent on getting it from the cytoplasm.
So for, 50 years plus, scientists have been looking for the doorway, the transporter?
You got it. Searching for that specific protein. Lots of candidates popped up over the years. GLUT transporters, ABC transporter.
But none of them were quite right.
None could be definitively proven. It was this stubborn knowledge gap, as some called it. Just an open question hanging out there.
Okay, so where does SLC35B1 fit into this picture initially?
Well, there were hints. Genetic studies had linked variations in the SLC35B1 gene to certain diseases, these congenital secretory stress syndromes.
Suggesting it was important somehow.
Yeah, important for the ER under stress, definitely. Yeah. But its actual job, that was fuzzy. It was classified broadly as a nucleotide sugar transporter, an NST.
So, moving sugars with nucleotides attached, not ATP itself.
Exactly. So its role in ATP transport was, maybe suspected by some, but unconfirmed, debated even.
And finding the real mechanism isn't just ticking a box, is it? It matters for bigger things.
Oh, hugely. ER energy levels control so much. Antibody production, how cells respond to stress, like in diabetes, that's the unfolded protein response, or UPR.
Even how viruses assemble.
Yeah, building viral glycoproteins. Yeah. It all depends on the ER having enough power, so figuring this out is fundamental.
All right, so half a century of mystery. These researchers decided to tackle SLC35B1 head-on.
Yep. Hypothesis. Despite the old classification, this is the ER's ATP transporter.
Yeah.
And they went all out to prove it, using multiple lines of evidence.
Very rigorous approach. Where did they start?
Well, first things first, does it actually bind ATP specifically? Basic question, but crucial.
How do you test that?
They use a few methods. One was thermal shift assays. Basically, you see if the protein gets more stable, less likely to unfold when something binds to it.
And did it with ATP?
Big time. When they added ATP, or ADP, the purified SLC35B1 got significantly more stable. Its melting temperature went up by over 5 degrees Celsius.
Wow. What about the things it was supposed to transport, like that sugar nucleotide?
UDP galactose.
Yeah.
Hardly any effect. Minimal stabilization. Same for AMP, the single phosphate version. That really pointed towards ATP and ADP being the preferred partners.
Okay, strong preference. Did they double check that binding?
They did, yeah. With a fancier technique, STD NMR spectroscopy. It directly spots the interaction between the protein and the molecule. Strong signals for ATP and ADP. Weak signals for UDP galactose and AMP. It just reinforced that binding specificity. Nailed it down.
Makes sense. So it binds ATP. Did they look at competition? Like, can unlabeled ATP block the binding of labeled ATP?
Exactly. Competitive inhibition assays. They use proteoliposomes, basically artificial bubbles with the transporter stuck in them.
Right.
And yes, cold ATP or ADP strongly competed away the signal from radio-labeled ATP-ADP. The numbers, the IC50 values, showed really tight competition, especially for ADP.
Interesting. What about other nucleotides?
That's where it got even more specific. Other similar molecules, CTP and UTP, also competed pretty well.
Oh, okay.
For GTP, competed very, very weakly. That suggested something interesting about the shape or perhaps the chemistry of the binding site. Maybe it prefers less hydrophilic bases, something we can come back to with the structure.
Okay, so binding dot check, specificity dot check. But the real proof is transport, right? Does it actually move ATP across the membrane?
That's the crucial step. So they used those proteoliposomes again. Purified SLC35B1, put it in the artificial membrane, isolated system, no confusion about what's doing the work.
Smart.
Robust transport.
Yeah.
If they preloaded the inside of the vesicles with ADP, they saw clear uptake of labeled ATP from the outside.
And the reverse?
Yep. Preload with ATP saw ADP uptake. It confirmed it acts as a genuine ATP-ATP exchanger, trading one for the other.
And did the speed, the kinetics match up with what you'd expect in a real cell?
It did, remarkably well. The measured came for ATP that's sort of its binding affinity during transport was around 3.4 micromolar.
Which is close to.
Close to the 3.5 micromolar reported years ago for ATP import into rough, crude ER preparations. So it fits the physiological picture.
And it was fast enough?
Oh, yeah.
Yeah.
The turnover rate, the KCAT, was about 12 ATP molecules per minute per transporter. Way faster than major ATP consumers in the ER, like the chaperone BiP. It can definitely keep up with demand.
Okay. Biochemical proof seems solid. Binding, transport, kinetics. But what about in a living cell? Does it matter there?
The ultimate test, right. They looked at cell growth.
How?
Using a massive CRISPR-Cas9 genetic screen in human cells. Basically knocking out genes one by one to see which ones are essential for survival.
And SLC35B1 showed up?
Near the top. It was one of the top 5 most essential SLC transporters for the cells to just survive and multiply.
Wow. So not just a niche player, a core housekeeping gene.
Absolutely. That confirmed its critical role in living cells beyond the lab bench tests. Cells really need this thing working.
So pulling it all together, the biochemistry, the kinetics, the genetics, it really seals the deal, doesn't it?
It absolutely does. SLC35B1 is the ER-ATP-ADP exchanger. The 50-year mystery solved. And that old classification as a nucleotide sugar transporter.
Debunked. History.
Pretty much. A really satisfying piece of detective work. But, finding the culprit is one thing. Understanding how they did it, the mechanism.
Yeah.
That often leads to even bigger surprises.
Right. The how, which means we need to see the structure, the atomic details.
Exactly. And that brings us to the next challenge, actually getting a picture of this protein.
Which I imagine wasn't easy. You said it's small for cryo-EM, the main technique for this now.
Very small. Around 35 kilodaltons. That's tiny for cryo-EM. It makes it really hard to get a good signal to align the images properly.
So how did they overcome that?
A really clever trick. They engineered an antibody fragment called FVMBP that binds specifically to SLC35B1.
Like putting a big handle on a small object.
Kind of, yeah. This fiducial marker made the whole complex bigger and more visible to the cryo-EM without messing up the transporter's function too much. A technical masterstroke, really.
Ingenious. So what did the first structure look like? What did they see?
Okay, so first the overall fold. It adopts what's called the drug metabolite transporter, or DMT fold. Pretty common fold. 2 bundles of 4 transmembrane helices each.
Okay.
But the state they caught it in was new for this family. It was open to the cytoplasm, an inward-facing state, a big cavity ready to accept ATP from the cell's main compartment.
So seeing that inward open state was a first.
For this specific family, yes, experimentally observed. They also saw something unusual in one of the helices, TM4, a helix break helix structure, like a flexible joint right in the middle.
Flexible. That might be important for movement later.
Good thought. Definitely hints at dynamics. And while the cytoplasmic side was open, the other side, facing into the ER lumen.
Sealed shut.
Sealed shut. By specific hydrogen bonds between conserved amino acids, like a gate, ensuring things only go through the proper pathway. E33 and N290, R37 and N109 doing the job there.
Okay, standard gating on that side. But the binding site for ATP inside that cytoplasmic cavity, was that standard?
Not at all.
Yeah.
This was maybe the biggest structural surprise. ATP and ADP didn't bind way down deep in the pocket like you see in many transporters.
No. Where then?
Only part way down, kind of perched halfway into the cavity. And the shape it adopted was weird.
Weird how?
Toroidal was the word they used. Bent. The N-phosphate group, the gamma phosphate on ATP or beta on ADP, actually arched back towards the adenine base.
Bent back on itself. That is unusual. How did the protein hold on to it in that bent shape?
Okay, so the phosphates, being negatively charged, were coordinated by positively charged residues. Arginine 276, lysine 277, lysine 120. These guys grabbed the phosphates. Q254 interacted with the sugar part.
Makes sense. Charge attraction. What about the adenine base part?
That was the other really strange part.
Yeah.
Usually you see specific hydrogen bonds or polar interactions holding the base. Here, not really.
No. Then what?
It was nestled into a hydrophobic patch. Greasy residues like isoleucine 257, valine 261, cysteine 269 formed this pocket that cradled the adenine base.
A hydrophobic pocket for the base. Ah, does that explain the competition results? Why GTP didn't compete well?
Exactly. GTP has a more polar hydrophilic base. This hydrophobic patch prefers the less polar bases of ADP, ADP, CDP, UTP. It just fits better. The structure perfectly explained the biochemistry.
That's elegant. Structure explaining function. Did they confirm those residues were important, like mutate them?
Oh yeah, standard practice,
Mm.
but crucial. They mutated those key residues, the lysines, the arginine, the hydrophobic ones, changing them to alanine, for instance.
And the result?
Transport activity plummeted, either severely reduced or completely gone. It really solidified the importance of this unique binding site, with its positive charges for the phosphates and that weird hydrophobic patch for the base.
Okay, so they know what it is, they know what the inward-facing state looks like, how it holds ATP, but how does it actually move it? That's the core function.
Right, the translocation mechanism. And this is where the standard models didn't quite fit.
Why not? What's the standard model again?
The rocker switch. Imagine the two bundles of helices rocking like a seesaw. One side opens, substrate binds, it rocks. The other side opens, substrate leaves.
Okay.
But with ATP only bound partway down in SLC35B1, if the helices just rocked inwards in the standard way,
Collision.
physical clash, the gating helices would literally bump into the bound ATP. It wouldn't work. It couldn't transport it.
So it has to be doing something different. How did they figure out what?
They needed to see the other side of the cycle, the outward-facing state, open to the ER lumen. This was tricky.
How'd they manage it?
A clever mutation. Changing one of those gating residues we mentioned earlier, E33 to alanine, E33A, subtly shifted the protein's preference and made it more likely to be caught in the outward-facing state.
Ah, so they could get snapshots of both the start and sort of the end point.
Exactly. They captured the ER luminal-facing conformation.
Yeah.
And comparing the two, that's when the really novel mechanism became clear.
Okay, moment of truth. What did the comparison show?
It showed that the adenine base, the core part of the ATP, moved vertically by about 6.5 angstroms as the protein switched from inward to outward facing.
Vertically. So not just rocking, but lifting or pushing.
Yeah, ratcheting is the terms they used. It's like the protein takes the ATP in one position and then actively moves or steps it to a different position further on the path, a stepwise movement.
Wow. So it's not just alternating access, it's repositioning the cargo mid-transport.
Precisely. Those gating helices, TM8 and TM9, swing inward to close the cytoplasmic side, as expected.
All right.
But that flexible helix we mentioned, TM4 with its brakes and key lysines, it keeps adjusting, kind of moving with the nucleotide to maintain contact as it gets repositioned.
And you mentioned a pivot point.
Yeah, that residue, R276 and TM9, seems to act like a hinge or pivot for the big movement of the gating helices during this step.
So in the outward state, where is the ATP-ADP now?
It's in a more canonical position, deeper in the cavity, ready for release into the ER lumen. They also proposed how ADP might enter from the ER side. Maybe the phosphates bind first, due to charge, and then the adenine base flips into that hydrophobic patch.
So it's a flip and step kind of thing.
Seems like it. And by combining multiple structures, they got 7 distinct snapshots. Empty, ATP-bound, ADP-bound, occluded states where it's trapped inside.
7. That's amazing detail.
Yeah, plus using advanced computational analysis called 3DVA on the cryo-EM data, they could piece together a movie, essentially.
Yeah.
A model of the entire stepwise transport cycle for both ATP going in and ADP coming out.
So this isn't just a variation on a theme, it's a fundamentally new way for an SLC transporter to work.
It appears to be, yes, a previously undescribed model. It suggests that for big, awkward molecules like nucleotides, which are amphipathic, part water-loving, part water-fearing, the transporter might need these intermediate binding spots, multiple steps to hand it off across the membrane.
Could this apply to other transporters too?
They suggest it might. The mitochondrial ADP-ATP carrier, SLC25A4, is another complex one thought perhaps to use a stepwise mechanism. So this provides a potential template. It's a new paradigm.
Paradigm shift. That's huge. It really underscores the innovation here. So let's talk about that broader impact. The first major innovation is obvious, right?
Solving the cold case. Finally identifying SLC35B1 and figuring out its basic job after 50 years. That alone is massive for cell biology.
But the way they did it was also innovative, the technology.
Absolutely. Getting atomic detail of a full transport cycle using 7 cryo-EM states, that's technically superb, a real tour de force.
And you mentioned that special grid technique.
Right. The graphene oxide grids developed by the graduate student Ling Z Chen. That was apparently key to stabilizing this tricky protein for imaging. Sometimes those technical hurdles are where the real breakthroughs happen.
Enabling the science. And they didn't just stay in the test tube, right? The live cell sensor.
Yeah, Maria Russa's split luciferase sensor, directly showing ATP import in living cells using this transporter that really bridges the gap, confirms the physiological relevance beautifully.
Plus the strategic choice by the PI, Charlie Yan.
To go for the human protein, the complex glycosylated one, not an easier bacterial version. High risk, high reward. And it paid off, giving results directly relevant to us.
So beyond the tech, the conceptual advance is this stepwise mechanism.
Definitely. A new transport paradigm, extending how we think about alternating access, especially for challenging substrates like nucleotides. And like you said, it provides potential blueprints for drug design, for inhibitor screening.
And it reframes how we think about the ER's energy supply, doesn't it?
Totally. It's not just some vague leak anymore. It's this specific choreographed ATP-ADP exchange, likely coupled to proton movement too, making it a symport. Much more precise and controlled than we imagined.
Which brings us to potential therapies. If this transporter is so crucial, could it be a drug target?
That's the exciting prospect. Understanding the mechanism reveals potential weak spots, druggable checkpoints. Maybe for diseases where the ER's protein folding system collapses, proteostasis failure.
Like in cancer? Or diabetes?
Potentially. You could imagine dampening down hyperactive ER in some tumors by targeting SLC35B1, since they need so much energy. Or maybe boosting it could help rescue cells struggling with the unfolded protein response in diseases like some forms of diabetes.
But there's a catch, right? ATP is fundamental.
Big catch. Messing with cellular ATP levels systemically is risky business. You'd need incredibly precise targeting. So, while the potential is there, it needs very careful consideration. It's a powerful dial to turn.
Still, having the blueprint is the first step. This work really lays a foundation for future studies on cellular energy, doesn't it?
Absolutely. It opens up new questions about how the ER's ATP supply is regulated, how it talks to the mitochondria, the main power plants. It's a framework for a whole new chapter in metabolism research.
Okay, so let's wrap up this deep dive. We've seen how this decades-old ER-ATP mystery finally got solved.
Yep. Human SLC35B1. Confirmed as the essential ATP-ADP exchanger for the ER. No longer a nucleotide sugar transporter in our textbooks.
And the mechanism. This completely novel, stepwise, ratchet-like movement. Just remarkable. It's a huge win for basic science and really a showcase of scientific ingenuity.
Incredible stuff, and it leaves you thinking, doesn't it?
It really does. We focused on how the ER gets its ATP via SLC35B1, but the cell's main ATP factories are the mitochondria. It makes you wonder, how tightly linked are these processes?
Is there like a direct line or a constant conversation between the mitochondria churning out ATP and the ER demanding it via SLC35B1?
Yeah, is it coordinated in real time to make sure the ER always has just enough energy but not too much? How does the cell balance that?
And what happens when that communication breaks down? Could glitches in that mitochondrial ER energy link contribute to metabolic diseases?
Right.
Or maybe even help tumors grow by messing with energy supply?
That coordination, that potential crosstalk, it feels like a whole new frontier just opened up, doesn't it? Lots more digging to do there.